Good morning. Today I will share with you a lipid metabolism. Let's see the outline of my discussion. We will start an overview of lipid, then we'll move to lipid injection, or we call that one cell lipolysis, and we'll move again into beta oxidation to acetyl CoA and fatty acid oxidation. Now let's see lipid injection. Lipids are available to the body from three sources that can be ingested in the diet, stored in adipose tissue of the body, or synthesized in the liver. Fat ingested in the diet are digested in the small intestine. The triglycerides are broken down into monosaccharide and free fatty acid, then imported across the intestine and mucosa. One across the triglycerides are synthesized and transported to the liver or adipose tissue. Fatty acids are oxidized through fatty acid or beta oxidation into two carbon acetyl CoA molecules, which can then enter the Krebs cycle to generate ATP. If excess acetyl CoA is created and overload to capacity of the Krebs cycle, the acetyl CoA can be used to synthesize ketone bodies. When glucose is limited, ketone body can be oxidized and used for fuel. Excess acetyl CoA generated from excess glucose or carbohydrate ingestion can be used for fatty acid synthesis or lipogenesis. Acetyl CoA is used to create lipid or lipid, a triglyceride, steroid hormone, cholesterol, and bile salt. Lipolysis is the breakdown of triglyceride into glycerol and fatty acid, making them easier for the body to process. Then let's see the main type of lipid. Lipid consists of triacylglycerol, triglyceride, phospholipid, and sterol. Let's see the structure of this lipid. This is the structure of fatty acid. This is esteric acid. And the upper side is the phospholipid. If we see this phospholipid, we have in this part the glycerol moiety. The glycerol consists of three hydroxyl group attached in each carbon. Then this hydroxyl group substituted by fatty acid group in this position, then the other one substituted by this group, phosphoryl group. Then if we see the space of filling a model of this lipid, we can see this phospholipid molecule. This phospholipid molecule consists of these two fatty acid group, and this one is a phosphate group, and this one is glycerol moiety. In the downside is a fatty acid molecule. Then if we see or have a look to the generic simplified of depiction of this lipid, we can see that this is as a head of the phospholipid, and this one is a hydrophilic, and this part is a hydrophobic, non-polar fatty acid tail. Then have a look to this bilayer. You can see that this kind of bilayer consists of this head and this is the tail. And if we consider this one as a missile, then this head will surround in the layer of this missile. Then let's move to lipid as an energy reserve. Lipid function primarily as an energy store in our body. Fat is body most concentrated source of energy. It's about 9 calorie per gram versus 4 calorie per gram for carbohydrate and protein. And typically 30 to 40% of calorie in human diet are from fat. Let's see this diagram. If we consider this one block as 1 calorie per gram, then for carbohydrate, we have 4 block. It means that a calorie per gram of this carbohydrate is 4 calories, and the same also for protein. This for protein also 4 calorie per gram. But have a look for this fat, it's almost 2 times compared to the protein or carbohydrate. The fat contain 9 calories per gram. Now let us see lipid as an energy reserve. Amount of lipid stored as energy reserve exceed the energy stored by glycogen from carbohydrate metabolism. You can see this one. The energy stored in our adipose tissue would enable humans to survive starvation for a month. Let's see this triglyceride. The source of energy is about 100,000 kilocalories, and the triglyceride store is about 15,000 grams. Compared to this glycogen, it's a small amount compared to this triglyceride. It's about 200 for liver and 400 for muscle only. Then for glucose, it's a bit smaller than glycogen, just only 20 grams, and the energy served is about 40 kilocalories. Then let's move to lipid function. Lipid function as energy storage. It means that this lipid incorporate into adipose tissue or fat tissue. And the second one, regulating and signaling. For example, triacylglycerol regulates constant temperature in the human body and regulate the hormone production. And the next is insulting and protecting. Visceral fat surround organ. Subcutaneous fat insulate the body underneath the skin. Let's see this figure. This is visceral, and then this one is a subcutaneous. And the other function of lipid is a transporting. Fat soluble vitamin require fat for effective absorption. Then let's move to lipid metabolism as an overview. Lipid metabolism are connected to the glucose metabolism pathway through acetyl-CoA formation in glycolysis and beta oxidation process. Let's see this acetyl coenzyme A, and then this is glycolysis, glucose converted into pyruvic acid, then this pyruvic acid will be converted to acetyl-CoA. The same thing that this lipid will be converted to fatty acid, then will be oxidized by using beta oxidation to get acetyl-coenzyme A. Then the same certain amino acid can be converted also to acetyl-coenzyme A, and also a ketone body. Then we can see from this row, this green row explain the lipolysis, and the red row is lipogenesis. Now let's move to lipid metabolism as an overview. Several pathways are involved in lipid metabolism. The first one is lipolysis of dietary lipid, and then fatty acid oxidation or beta oxidation, ketogenesis, ketone body oxidation, cholesterologenesis, and lipogenesis. Let's see from the upper side of this one. This is lipogenesis, and then beta oxidation to get acetyl-CoA. Then from carbohydrate and amino acid, it can be also converted to acetyl-CoA. From this acetyl-CoA, we can see that this is lipogenesis, and also the fatty acid can be converted from the D. And after we got this one acetyl-CoA from this pathway, then this acetyl-CoA will enter citric acid cycle to get the NAD. Now let's look to lipid injection, or we call that one as a lipolysis. There are 4 types of lipase: lingual lipase, gastric lipase, pancreatic lipase, intestinal lipase. The most active is pancreatic lipid. Lipid or lipid are digested in the small intestine. A lipid absorption occurs according to the lipid type in human body. That's it. This one, this is triglyceride, and will be converted into glycerol and fatty acid. The first one, this triglyceride will be emulsified by bile salt and converted by hydrolysis using enzymatic hydrolysis. For this, phospholipid will be converted by phospholipase and converted into lysophospholipid. And the other one is a cholesterol ester. This one will be converted by cholesterol esterase to fatty acid and free cholesterol. Now let's have a look to this quiz for lipid in blood. According to Lipoprotein, are lipid generally soluble or insoluble in the aqueous nature of blood? I'll repeat it again. According to lipid property, are lipid generally soluble or insoluble in the aqueous nature of blood? Then the answer for this is lipid are generally non-polar, therefore not very soluble in blood. There is a polar end in the lipid structure which makes them slightly polar. Now have a look to this lipoprotein as lipid transport mediator. Let's see this figure. This is apoprotein. This one of phospholipid, cholesterol, and this one is a lipid core. Then the amphipatic nature of phospholipid inhibit lipid transport in body fluid due to lipid-water incompatibility. And for this lipid transport mediator, its special particle is needed to facilitate lipid transport. Lipoprotein are a lipid plus a protein consists of an inner core of hydrophobic lipid surrounded by a surface layer of phospholipid, a cholesterol, and a polypoprotein. Let's see this lipoprotein as a lipid transport mediator. Plasma lipoprotein can be divided into 7 classes based on size, lipid composition, and apolipoprotein such as kilomicron, silomicron, VLDL, IDL, LDL, HDL, and LPA. Silomicron, 10 mol, VLDL, IDL, and LDL, and LPA are all pro-atherogenic, while HDL is anti-atherogenic. Apolipoprotein have 4 major functions include 1. Serving as a structural rule, 2. Acting as a ligand for lipoprotein receptor, and 3. Guiding the formation of lipoprotein, and 4. Serving as a activator or inhibitor of enzyme involved in the metabolism of lipoprotein. So you can see from this figure, this is the classification of the lipoprotein. This is silomicron and silomicron remnant, and this FLDL, IDL, LDL, and HDL, almost all in the liver and a small intestine. There are always a relatively constant supply of lipid in the blood, although the concentration increases immediately following a meal. A lipid in the blood are absorbed by liver cells to provide energy for cellular function. If lipid level in the blood become too low, the body synthesizes lipid from other food such as carbohydrate, or remove lipid from storage or from adipose tissue. Excess of excess of this lipid in the blood are eventually converted into adipose tissue. Immediately after absorption of lipid, there is turbidity in the blood plasma due to circulating silomicron. This is approximately 2 hours after meal, and turbidity is soon cleared by lipoprotein lipase. Let us see this one. This is normal plasma, and this is lipemic plasma. Lipemic plasma, it means that there is a turbidity in the blood plasma due to circulating silomicron. Now let us see the lipid injection summary. Let's see from the upper side of this figure. We'll start from this fat ingestion indeed will enter stomach and will pass by into the small intestine. Then we will see the pathway or step by step of the mechanism of entering this fat ingestion indeed into this system. The first one is bile salt emulsify that fat in the small intestine, forming mixed micelle. And the second one, intestinal lipases degraded diacylglycerol. And the third one, fatty acid and other breakdown products are taken up by the intestinal mucosa and convert into diacylglycerol. And the fourth one is that diacylglycerol are incorporated with cholesterol and apolipoprotein into kilomicron. And the next one is kilomicron move through the lymphatic system and bloodstream to the tissue. And the next one, the lipoprotein lipase activated by ApoC8 in the capillary convert diacylglycerol to fatty acid and glycerol. And then this fatty acid enter the cell. And the last is fatty acid are oxidized as fuel or re-esterified for storage. So from this summary, we can see that step by step entering of this fat into the digestion system. Now let's see abnormal level of lipid in blood. Abnormal high level of triglyceride and cholesterol are thought to be involved in hardening of the artery. Lipid may be deposited in the wall of artery as a partial consequences of their insolubility in the blood. Let's see what happened with this one. This is HDL and this is LDL. Let's see, this LDL it's bad and this is HDL is a good. So in discussing LDL, we can see that this LDL and this LDL will deposit in this capillary blood system, and somehow it will block this capillary system. Then have a look to this lipid profile test. We can see that there are some measuring of a parameter of this lipid profile test. For example, total cholesterol, and this profile also divided into optimal, intermediate, and high. For example, for total cholesterol, there are also at least two units. We can use milligram per deciliter or millimole per liter, and so on. So if we see that total cholesterol for optimal is about 200 or less than 200, and for high is more than 259. Then if we see that this LDL cholesterol, this is calculated, this is less than 130, and for high is more than 159, and for triglyceride it's about less than 150, and for high profile is about 200. I mean more than 199. So let's move to fatty acid oxidation or beta oxidation. Beta oxidation is the process by which fatty acids are broken down to produce energy. Regulate the cell requirement with the stimulation by epinephrine and glucagon. In this case, require NAD plus and FDA. The enzymes that participate in the fatty acid catabolism are located in the mitochondria. Let's see this one. The diagram of converting fatty acid into a small molecule by oxidation, or we call that one as a beta oxidation. Then let's see what happened with this fatty acid oxidation. Initiation. This initiation occurs in the outer mitochondrial membrane. The relative inner long chain fatty acids must first be activated by coenzyme A using fatty acid coenzyme A as enzyme. For each molecule of fatty acid activated, one coenzyme A and two ATP are used. Let us see this equation. We have in hand this fatty acid will react with coenzyme A and convert it to fatty acid coenzyme A. In this reaction, it's need energy, ATP, and this ATP will convert it hydrolyzed to AMP, conversion into AMP, not ADP. Let's move to this transport of fatty acid coenzyme in mitochondria. Let's see for the first one. This is the schematic of mitochondria. This is a matrix and this is a cytosol. From this one, we can see how come that this carnitine enter into cytosol as a carnitine by using this mechanism. This is a transport of fatty acid across the membrane. This fatty acid coenzyme A is converted into fatty acid carnitine by carnitine using the help of carnitine acyl transferase 1. Fatty acid carnitine pass into the inner membrane of mitochondria. The medium and short chain fatty acid are carnitine independent. Now let's move to fatty acid oxidation state. This set occur in mitochondria matrix and also called as beta oxidation, since the beta carbon undergo oxidation to a carbonyl group and produce different outcome according to the structure of the fatty acid. And this state also connected to citric acid cycle and electron transfer chain. We have a look this one. Then pathway. Fatty acid CoA is oxidized through a sequence of 4 reactions in the beta carbon. This process occurs continuously until 2-acetyl CoA molecules are produced in the final step. So this will be continuously occur until 2-acetyl CoA molecules are produced. Let's see this one. First step. The first step involves acetyl CoA dehydrogenase, and this enzyme has 3 isoenzymes. It catalyzes different type of fatty acid, long, medium, and short chain. A trans double bond is formed on fatty acid CoA by removing 2 electrons. In this case, let's see this one. This is acyl CoA and will be converted into trans delta 2 enoyl CoA, and in this case, this FAD will be reduced to FEDH2. Second step. The second step is The enzyme is acyl-CoA hydratase. The process is hydration step in the double bond between alpha and beta carbon, resulted in addition of hydroxyl group to the beta carbon and proton to alpha carbon. In this second step, no energy is involved. Let's see the structure of this equation. This is trans delta 2 enoyl CoA and will be converted into L, this one L3 hydroxyacyl CoA. Let's see, you should remember that this configuration should be L. Then let's move to the third step. Enzyme involved in this step is beta hydroxyl acyl CoA dehydrogenase. The process is one electron and two protons are removed from the hydroxyl group and beta carbon is oxidized, produce a molecule of NADH. In this case also, this process is reduction process. In this one, NADH will be converted into NADH to get the ketoacyl CoA. Final step of this fatty acid oxidation, the enzyme that involved in this final step is beta ketothiolase, and the process is thiolytic reaction involve cliff of block between alpha and beta carbon. And procedure is as one acyl CoA and a fatty acyl CoA with two carbon shorter. Let's see this one, 3 ketoacyl CoA, then will produce two molecules, one molecule of acetyl CoA and this acyl CoA. In discussing this acyl CoA, two carbon atom shorter than this 3 ketoacyl CoA. Now have a look to this fatty acid spiral. The beta oxidation process will be repeated until the final step result in the two molecule of acetyl CoA. Thus, making this pathway is also called fatty acid spiral. This is, let's see this one. Come up from the C14, then will produce acetyl CoA, shorten it into carbon of 12 and produce again acetyl CoA till the end will get acetyl CoA. Then we call this one as a fatty acid spiral. Let's move to this one. Researcher working with isolated mitochondria in the early days actually did find ratio of 25 and 15 experimentally, but did not believe this could be the real power. They were operating under the assumption the oxidative phosphorylation produce ATP by a mechanism similar to substrate level phosphorylation, as is seen in the GTP dehydrogenase reaction of glycolysis. The chemical intermediate hypothesis required integer number, so they rounded up to 3 and 2. Now let's see the fatty acid, the energy balance of this one. For example, of this one is 1 × 10⁻⁴ palmitic acid. This lipid is saturated fatty acid. This is 2 rounds of oxidation and will get acetyl-CoA produced. And in this oxidation, it will produce 2 ATP used for activation step. I'm sorry to say, in this case, it will be used 2 ATP for activation step. For each round of oxidation, 1 NADH equal to 2.5 ATP, 1 FADH2 equivalent to 1.5 ATP, 1 acetyl-CoA will enter Krebs cycle. Then in Krebs cycle will produce 1 GTP, 3 NADH, and 1 FADH2. Overall net yield of this reaction is 106 ATP per C16. Now let's have a look to the fatty acid oxidation special cases. The first one is unsaturated fatty acid need isomerization and reduction step. And the second, odd chain length fatty acid is in the final step will produce acetyl-CoA and propionyl-CoA instead of 2-acetyl-CoA. And pyruvate chain fatty acid, first beta oxidation occur in peroxisome before transported into mitochondria. Then the branch chain fatty acid will degrade by alpha oxidation, generate formyl coenzyme A and propionyl coenzyme A along with acetyl-CoA. I think this is a kind of interesting knowledge considering the lipid. Then thank you so much for your attention. See you then at the same time, at the same moment, and we will discuss the next lipid metabolism part 2. Thank you so much.